So here we are, the big reveal, the end of glucose metabolism, second and last chunk of the Lecture 16 series. The rest of Chapter 20, we've already talked in the first chunk about the electron transport chain briefly, reminding everybody of what we did there and why it was important. And we talked about more theoretically and the larger kind of wide view of linking that electron transport chain action, that proton gradient, to oxidative phosphorylation. But here we will actually describe the process of ATP production. How does the protons flowing through ATP synthase actually create ATP? And we'll end with an overview of all of glucose metabolism, focusing exclusively on the ATP yields of those processes. So, I want you to kind of reflect on a hydroelectric power plant. How many of you know how a hydroelectric plant works, what it is and how it works? This is an example of a hydroelectric plant. Hydroelectric plants are always near moving water, and they look like dams, except they are functional dams. They create electricity. So this is actually an electricity power plant. And if we were to take a look inside that dam, we would essentially see this setup. Here is the far side of the dam where the water is built up, and here is the low side of the dam where the water flows to. Obviously, the water wants to flow from where there's more of it to where there's less. But in order to do that, the water has to flow through this flume, and this flume is within the interior of the dam itself. The water flows through this flume, going from its high level to its low level, and as it goes through the flume, the water physically turns this turbine. And it's the turning of this turbine that generates electricity. Once the water makes it to the other side, it's no longer energetically active. We've released that kinetic energy, and no more electricity can be made. So it's key for a hydroelectric plant to always have much more water on the far side of the dam than on the low side, so that you have this water pressure pushing water through the flume to turn the turbine and generating electricity. We need that gradient of water. That's how a hydroelectric plant works. Oxidative phosphorylation works under the exact same principles. And I know I'm an analogy-based instructor. I know I use analogies quite a bit. No analogy here. ATP synthase is a turbine. It physically spins as protons flow through it. It's one of the most amazing things I've ever learned about in my life. We will be physically spinning the F0 region of ATP synthase as a turbine with protons flowing through it, and that spinning will drive the production of ATP. Essentially, the mechanical energy generated by protons flowing through the F0 region of ATP synthase will allow there to be spin energy or torque energy that will drive the production of ATP. And I want you to stop and take a moment to think about this. We, people, humans, developed hydroelectric plants somewhere between 150 and 200 years ago. And cells had been doing the very, very same thing for millions of years. Incredible. It's just incredible that this happens inside our own cells. It's happening inside your own cells right now. Right now, each of you sitting there listening to this lecture have trillions of hydroelectric power plant turbines spinning in your mitochondria, generating ATP for yourself. Just incredible. This idea that I'm sharing with you for ATP production was originally thought of as chemoosmotic coupling. The idea here is that a chemical gradient, specifically a chemical gradient of protons, was coupled to the production of ATP. So that's what that name is meant to capture. Chemoosmotic coupling states that the proton gradient and the movement of protons across the inner mitochondrial membrane, that was the proton that we generated by the electron transport chain, was coupled to ATP production. Now, that's somewhat true. There's no doubt that the proton gradient is important. Look at all the trouble we went through to create it. Everything done in the electron transport chain was devised around releasing small packets of negative delta G energy solely for the purpose of pumping protons. So the gradient is important. Pumping protons is what we did with the energy from glucose. Additionally, there are 4 more specific lines of evidence that support chemoosmotic coupling. Again, by chemoosmotic coupling, we just mean that the proton gradient makes ATP. The first line of evidence is that the cell, or the mitochondria, is really nothing more than a system of closed compartments. We have the matrix, the inner mitochondrial space. There's an inside and an outside where the proton gradient can be controlled and maintained. If the proton gradient wasn't important, why would mitochondria evolve in this way in order to be able to segregate protons? Why would this happen if not for the need of a proton gradient? 2, experimentally, we can isolate bits and pieces of inner mitochondrial membrane that have ATP synthase in them, and we can close those bits of inner mitochondrial membrane up and manipulate pH, manipulate proton concentration gradient, and we can actually get ATP to be made. We can create these kind of artificial ATP synthase membranes, and as long as we have a proton gradient, as long as we have a differential pH, these things will pump out ATP for us. What that tells us is that ATP production, oxidative phosphorylation, whatever you need for that, relies on pH, a proton gradient, and this stuff that you're looking at here, ATP synthase. 3, if experimentally you can establish a proton gradient, again by manipulating pH values on either side of ATP synthase, and you have a membrane in between those differential pHs that contains the protein ATP synthase, you'll make ATP. That tells us that protons flowing through ATP synthase allows ATP to be made. And fourth, the fourth line of evidence that supports chemoosmotic coupling, there is indeed a pH gradient in mitochondria. We have developed the technology to physically measure the pH of the inner mitochondrial space and the mitochondrial matrix. And indeed, the inner mitochondrial space is far more acidic, which means a much higher proton gradient than the mitochondrial matrix. So there is a pH gradient in these organelles. It's real and it exists. So we have a system where the inner mitochondrial membrane is a very strict membrane. Almost nothing will get across it without a channel. We have protons that want to get across it but can't because they lack a channel. That's the property of the inner mitochondrial membrane that maintains the proton gradient. But as we've already said, if we're going to harvest the potential energy of those protons, we need to convert that potential energy into kinetic energy. We have to get those protons to move, and the only way they're going to move across the inner mitochondrial membrane is through a channel. They need something to flow through. Just like if this water was going to get from one side of the dam to another, they had to flow through the flume. Now, once we have that kinetic energy of motion going, once that water is going to flow, why not put something in its way that the water can spin, generating energy for us, generating power? And the same again for the protons. If the protons are flowing through ATP synthase itself, why not use that energy of proton motion to power the production of ATP? Again, ATP synthase is a large protein complex, large enough to see on a scanning electromicrograph, and it is, among many other things, a proton channel. The F0 region of ATP synthase allows protons through it. So a nice schematic view of ATP synthase. Here is that F1 region, here is the head region, where we will convert ADP into ATP. The protons are flowing through the inner mitochondrial membrane by going through the F0 region, the transmembrane region of this beautiful complex. So, if you think about it, we just achieved the coupling. If we're talking about chemoosmotic coupling, we're saying that the flow of protons drives ATP production. If protons are flowing, you get ATP. If protons are not flowing, you cannot get ATP. That's what chemoosmotic coupling states. And we just coupled proton flow to ATP production. ATP synthase itself is that link. Protons moving is coupled to making ATP through ATP synthase itself. We can see it right here on this diagram. Here's the movement of protons in the F0 region. Here's the production of ATP in the F1 region, and they are physically linked. So we couple the movement of protons to ATP production in this one beautiful complex. Let's move to a refined version, or a refined way of thinking of ATP production, though, and we'll call this conformational coupling. Conformational coupling says that yes, the proton gradient is important, no doubt about that. But the proton gradient is not directly linked to ATP production. the proton gradient is indirectly linked to ATP production. You might remember a long time ago, I'm sure you do remember, we had the sequential model and the concerted model for describing allosteric enzymes. And remember we said that those were just two different ways of thinking about how allosteric enzymes work. This is the same idea here. Chemoosmotic coupling and conformational coupling are two different ways of thinking about how a proton gradient can cause ATP to be made. So conformational coupling is just another idea to describe ATP production in oxidative phosphorylation. So this is a theory or a model that's separate from chemoosmotic coupling. The idea of conformational coupling is that the proton gradient is important, but what it is important for is driving conformational changes of ATP synthase itself. In other words, conformational coupling says that when protons flow through ATP synthase, all that happens is that ATP synthase goes through conformational changes. And it is those conformational changes that result in ATP being made. It's a subtle difference, but it's a distinct one. Chemoosmotic coupling says the proton gradient causes ATP to be made. No proton gradient, no ATP. Conformational coupling says the proton gradient causes conformational changes to be made, and conformational changes result in ATP production. The implication in conformational coupling is that if you could somehow drive those conformational changes to occur without a proton gradient, you'd still make ATP. Remember, chemoosmotic coupling says no proton gradient, no ATP. Conformational coupling says proton gradient fine, but if you can get conformational changes without a proton gradient, you would still make ATP. The thing that is directly making ATP in conformational coupling are these conformational changes of ATP synthase. We now know, through many, many studies, that conformational coupling is the valid model. This is how ATP production works. Yes, the proton gradient is needed to make ATP under normal cellular circumstances, but the only thing that the proton gradient causes is a physical change or conformational change in ATP synthase. The protons flow through the F0 region of ATP synthase. The F0 region spins like a turbine, and that spinning drives conformational changes to occur in the F1 region of this complex. It is those conformational changes that drive ATP production. If you could experimentally get the F0 region to spin without protons, you would make ATP all the same. You can make ATP in this way without protons. And that's what makes conformational coupling distinct from chemoosmotic coupling. And that's what we say here. So what are these conformational changes? Let's finally tell the story of oxidative phosphorylation. ATP synthase, more specifically the F1 region of ATP synthase, has three different sites, three different subunits where substrate can bind. And they're shown here. 1, 2, 3. You'll notice that all three sites are in different conformations or have different shapes at any one time. In other words, you never see two subunits in the same state simultaneously. Substrate for making ATP is ADP, an inorganic phosphate. That's what we're going to start with. And each of these three sites can cycle through three different conformations each. There is the loose conformation, L, which binds the substrate, ADP, an inorganic phosphate. There is the tight conformation, T, which squeezes ADP and inorganic phosphate together until they spontaneously form ATP. And then there is the open site, or O, where that newly made ATP product is released back into the mitochondria. And that's it. That's the story. At any one given time, each of the three subunits are on three different states, loose, tight, and open. And as protons flow through the F0 region, and the F0 region spins, it drives each one of these subunits to cycle through loose, tight, open. So let's start by following just one site for simplicity's sake. This is just a larger version of the schematic I showed you before, but we're going to zero in on what's going on just here on this type site. We'll talk a little bit about what happens at these other subunits as well, but we're going to focus on what happens here. So in this state, this site that we're focusing on is in the loose conformation. And yes, this is in tight and this is in open, but we're going to direct our attention to here, loose. And the loose conformation binds substrate, substrate being ADP and phosphate. Protons now flow through the F0 region. And as those protons flow through the F0 region spins. And that spinning causes conformational changes to occur. Here is the stalk of the F0 region. We can see that stalk coming up here through the head region. See that? And this stalk has a little bump to it, a little perturbation. And here is that stalk. Imagine that we're looking down from the top. So imagine we're kind of bent over here and we're looking down onto the F1 region, and this is that stalk with the perturbation, that little bump there. Wherever that bump is going to be the open site. So protons flow through the F0 region. The F0 region spins. The stalk spins with it. And there goes the stalk, spinning as a result of protons flowing through the F0 region. Now remember, I told you, wherever that little bump is the open site. So this open site is going to shift down here. This loose site is going to shift down here. This open site is going to shift over here, and the tight site is going to shift upwards. Let's do that. So I just did that shift. Again, the substrate's going to stay put. All that's changing is the conformation. So this open unit is going to shift to loose. This tight unit is going to shift to open. And this loose unit is going to shift to tight. I'll do it again. And it shifted as a result of proton movement. Well, remember what the tight conformation does. The tight conformation squeezes that substrate into ATP. So ATP was just made. But protons are still flowing through the F0 region. So this thing continues to spin and spin. As those protons spin through the F0 region and that bump moves again, we are going to shift counterclockwise once more. This loose site will shift over to this subunit. This tight site will shift to where the loose is now. And the open site will shift up to the subunit we're focusing on. And there it goes. Remember, the role of the open site is to release ATP into the mitochondria. And the ATP we just made is released. And that's really the story. Just imagine, though, as protons flow, all of this happening in all of those subunits at the same time. That stalk of the F0 ATP synthase region constantly spinning and spinning as protons flow through the F0 region, as protons follow their gradient across the inner mitochondrial membrane, driving all sites to constantly flow or move through their conformations. Loose, tight, open, loose, tight, open. Bind substrate, squeeze it into ATP, release that ATP. Bind substrate, squeeze it into ATP, release that. And then another subunit is actually cycling at the exact same time, tight, open, loose, tight, open, loose. And the last one is open, loose, tight, because they're always out of sync. If you do that, you'll crank out a lot of ATP. And in fact, you do. So here's a better diagram of what's actually going on in oxidative phosphorylation, and I realize there's a lot going on here, so we're going to let this spin for quite a while, and we're going to see it a few different ways. I want us to first direct our attention again to this upper chamber here, this upper subunit, and let's just watch as it cycles through the three conformations. Loose here is a brightish red, tight is going to be orange, and open is pink. So loose, what binds? Substrate, ADP and phosphate. As we move next to tight, what happens to that substrate? It's made into ATP. And as we cycle to pink open, we release that ATP into the mitochondria. So bind substrate, squeeze it together, release. Bind substrate, squeeze it together, and release. That's happening in this top subunit. Now draw your attention a little bit further back and just take note of what's going on in the entire F1 region of ATP synthase. You'll notice that at any one time, none of the subunits are in the same state. One is always in loose, another is always in tight, and the third is always in open. But if we go to any one of these other subunits, we'll see that it's really telling the same story. Loose binds substrate, tight squeezes it together, and then ATP is released. Bind, squeeze, release. Bind, squeeze, release. If we come over here, we see that the same thing is occurring. Bind, squeeze, release. ADP and phosphate binds, squeezed into ATP, and released. So what we're really doing is we're creating ATP in the same exact way at 3 different subunits, just out of sync with each other. For those of you who are familiar with the Row, Row, Row Your Boat song sung in a round, it's almost like ATP production is happening in a round. The first version is loose, tight, open. The next version is tight, open, loose. And the third version is open, loose, tight. But we're singing the same song, we're doing the same thing, and we're cranking out tons and tons of ATP. The last thing I'll point out is this is really trying to show this as accurately as possible. And so what's in black here is that stalk region of the F0. What's causing this conformational switch to occur is the spinning of this black region within the center of the F1 domain. And the reason why this is spinning is because it's connected to F0, and the reason why F0 is spinning is because protons are flowing through it. We created that proton gradient in the electron transport chain to have those protons flow through the F0 region of ATP synthase so that this can happen. Conformational coupling. The textbook says, in essence, the chemical energy of the proton gradient is converted into mechanical energy in the form of the rotating protein. This mechanical energy is then converted back into chemical energy stored in the high-energy phosphate bonds of ATP. Very true. We conducted chemical redox reactions that we use the energy to pump protons. We allow those protons to flow, driving the physical motion of ATP synthase, and ATP synthase then creates high-power energy molecules for us in the form of ATP. There's a video I'm going to show. I'm going to hope that this translates well through my office speakers here, and that you can follow along. It's about a 3.5-minute video, and it shows this same process in a different way, and I think it's important to hit this as many times as possible. So here we go.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins.
I'm going to pause it there for 1 second, because I don't want to call that other membrane-bound proteins. Come on, right? Don't insult our intelligence. We know what these are. This is complex 1, complex 3, and complex 4. And not only that, we even know what this is and this is. You say it out loud. Make sure you remember. This is CoQ, and this is cytochrome C. These are the members of the electron transport chain that deal exclusively with NADH plus H, and they were there to establish the proton gradient that we're about to leverage. Other associated membrane proteins. Come on.
Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
And I'm going to jump in here again and say that you can see what's actually happening in the context of what we've described. This subunit just shifted to the open conformation, releasing it. This one just shifted to the tight, making ATP.
The process has now completed, and the result is an equal number of protons.
And now we're about to shift here into the into the loose conformation so that substrate can bind. And as we do, this one is going to be released as open.
On each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
And so what she's saying there at the very end is that we can't spin ATP synthase any longer in this animation because the number of protons on both sides of the inner mitochondrial membrane have equilibrated. They're the same. And while that's true, she went on to say, well, that's never really going to happen in a mitochondria because these protons will be picked up and pumped right back out again. And we know how that's going to happen. That's going to happen right here. These are going to be responsible for taking the protons that flow in through ATP synthase and pumping them out again. We're going to pump protons in complex 1, 3, and 4. We're going to absorb protons when we reduce CoQ and when we reduce oxygen to water. So we'll constantly be reestablishing that proton gradient as we let protons flow through ATP synthase. What a beautiful, amazing system. On to the energetics. Now, we're talking about energy yields, but do remember that when we say energy here in this context, we mean ATP. That's really what we're after. And so this is the entire process that we've been discussing all along. Remember, starting with the very first step and third step of glycolysis, we actually burned 2 ATPs, but those are the only ATPs we did burn. The rest of the ATPs were made rather than used up. We made 2 ATPs later on in glycolysis. We made 2 more in the very last step of glycolysis. So glycolysis netted us a yield of 2 ATPs. We made 2 ATP molecules as well in the citric acid cycle. And why did we make 2? Remember, we are encompassing or accounting for everything we did with a single molecule of glucose. So a single molecule of glucose was converted into 2 molecules of glyceraldehyde 3-phosphate, which then went on to become 2 molecules of acetyl-CoA, which fed into 2 different citric acid cycles, each one of those citric acid cycles making us 1 ATP. So we actually make 2 ATPs from those 2 citric acid cycles of a single molecule of glucose. And then the rest of it comes down to the energy made by the electron transport chain and the proton gradient, and then oxidative phosphorylation itself. So most of the time, depending on how we get intermediates of glucose metabolism to the mitochondria, and that's what this refers to, we make somewhere between 30 and 32 ATPs for every single glucose molecule consumed. Amazing! If we were to do anaerobic respiration, we're just doing glycolysis, so we make 2 ATPs for every glucose consumed, a much less efficient process. And so before we wipe our hands of glucose metabolism, I think it's critically important that we just tell this whole story one last time because we now have a complete story to tell. We started with glucose and phosphorylated it to glucose 6-phosphate, isomerized it to fructose 6-phosphate, phosphorylated it again to fructose 1,6-bis-phosphate, and then we cleaved that into 2 molecules of GAP. Of course, isomerizing DHAP into our second GAP molecule. gap was turned into 1,3-BPG. We then allowed that to give up a phosphate group to make ATP for us, giving us 3PG. 3PG was mutased into 2PG, which we then converted into phosphoenol pyruvate. We lost that second phosphate group to 2 more ATPs, giving us pyruvate. That pyruvate was converted into acetyl-CoA by the pyruvate dehydrogenase complex, which involved converting that acetyl into hydroxyethyl, linking that hydroxyethyl onto TPP, swapping that hydroxyethyl onto lipoic acid, and that lipoic acid was then oxidizing to hydroxyethyl, becoming reduced itself, and oxidized hydroxyethyl is acetyl. Acetyl was transferred to CoA, giving us acetyl-CoA, and then we offloaded those electrons and protons from lipoic acid onto FAD and eventually onto NAD, and we know where they went at this point. These NADH plus H electrons and protons went directly to complex 1 of the electron transport chain. But it's the acetyl-CoA that we were interested in. The acetyl-CoA, or the acetyl group of acetyl-CoA, condensed with oxaloacetate, and from that point on went through the entire citric acid cycle. 2 successive rounds of oxidative decarboxylation caused us to lose carbon dioxide, but more importantly gain electrons, and 2 more redox reactions later on in the citric acid cycle gained us further electrons still. All of those electrons were transferred to NADH plus H, except for the electrons stolen by succinate dehydrogenase. Those electrons and protons were kept on FAD, part of the enzyme itself. The NADH plus H electrons are mobile, and NAD carried those electrons on to complex 1. Complex 1 pumped protons and moved those electrons on to CoQ. CoQ took those electrons and transferred them to complex 3. The electrons held by succinate dehydrogenase were also transferred to CoQ, and CoQ brings those electrons and protons to complex 3 as well. Eventually, complex 3 transfers those electrons to cytochrome C. Cytochrome C brings those electrons to complex 4, and complex 4 eventually uses those electrons to reduce oxygen into water. All along the way, with all of those changing hands of electrons, we are pumping protons into the inner mitochondrial space or absorbing protons from the matrix. The end result of the ETC is an abundant amount of protons outside of the inner mitochondrial membrane and far fewer protons inside. Those protons desperately want to flow, and flow they do through ATP synthase. As those protons flow through the F0 region of ATP synthase, they cause that F0 region to spin. That perturbation in the stalk of the F0 moves within the F1 head group and causes conformational changes to occur within the F1 region of ATP synthase. And those conformational changes follow a set pattern. They go from loose, where ADP and phosphate is bound, to tight, where those 2 molecules are squeezed into a single molecule of ATP, to open, where the ATP is released back into the mitochondrial matrix. And that happens by the bucketful. And so we go from 1 molecule of glucose to 30 to 32 molecules of ATP through this glorious and amazing metabolic system. In summary, ATP synthase allows the energy stored in the proton gradient to be manifest into the production of ATP. And it does so with protons flowing through the F0 region of ATP synthase. That's our proton channel. And as that F0 region spins, it perturbs or causes conformational changes in the head region of the F1 region of ATP synthase. Chemoosmotic coupling is the idea that the proton gradient and the proton gradient alone creates ATP. No gradient, no ATP. Conformational coupling, on the other hand, states that the proton gradient is indirectly linked to ATP production only because that proton gradient normally causes the conformational changes to occur in F1. If you could get those conformational changes to occur some other way, you'd still make ATP. And that is true. Conformational coupling more accurately describes how ATP is made through oxidative phosphorylation. From your textbook, they show this process in the same way here. You can see that we have the loose, tight, open schematic. I won't belabor it one last time, but there are diagrams in your textbook to show this as well. Hopefully I end with you as amazed as I always am with this story, just a truly amazing story. And take that one moment of mindfulness right now if you can to appreciate the fact that everything that I've described is not some abstract thing that occurs somewhere else in the universe. It is an amazingly unique life process that is occurring in you right now as you sit there.